Hi, the next zoonotic disease we're going to talk about is scabies. Scabies is caused by a mite, specifically one called Sarcoptes. And it's not a true zoonotic infection in the sense that there are multiple forms of the condition. So there are mites that infect our domestic animals, specifically dogs and cats. It's very common in those. And then there's mites that infect humans. And they are host specific, meaning that they can only reproduce on their host. So a dog mite can only reproduce on a dog, and a human mite can only reproduce on a human. However, these mites can live off of the host for as long as 48 to 72 hours. And essentially that means that your pets can give you scabies, but in this case it's referred to as pseudoscabies. So that is the zoonotic form of scabies that is acquired temporarily because of contact with an infected pet. The way that scabies is transmitted is it requires skin-to-skin contact and for a prolonged period of time. So it's not just shaking hands, it's not transmitted by fomites or vectors. It requires that you actually touch the person that has the disease for a period of time. So it's much more common in crowded conditions as a result of that. As you can see on the previous slide, it causes these little red sores that are extremely itchy or pruritic. And the itching is actually a direct result of the actions of the female mite and its attempt to reproduce in the skin. So the mite burrows into the skin to bury eggs, and it lays up to 3 eggs a day. And as a result of that, it also is consuming skin tissue and defecating in the skin. And it's actually the mite fecal material that's believed to be the antigen that results in the itching. The eggs hatch pretty quickly and are mature within 2 weeks. So it doesn't take long for a single mite to become a pretty heavy infection. But as I was saying, they can only live off the host for a couple of days, a max 3 days, and that's because they do require the skin cells in order to live and as a food source. It's relatively easy to diagnose scabies. Actually, it's done with what's called a skin scrape, and this is a sample, a small sample of skin that's acquired with a blade. There's a good picture of it on this slide. And you simply look for the mites under the microscope. It's believed that as few as 3 mites can cause severe itching. The disease is also very easy to treat. The big problem with scabies is it can lead to secondary skin infections because of the break in the skin, and those can be very problematic. For example, MRSA, because of the break in the skin. So the second option for discussion post has to do with this particular condition, and what I want you to research are what type of people, groups of people are more likely to get scabies than others, and how can you best prevent it. And so for this particular one, focus on the human-to-human transmission of scabies rather than the zoonotic form pseudoscabies. Right. So moving on, the third zoonotic disease we're going to focus on is toxoplasmosis. This is related to the earlier one we discussed, and it's also a protozoal parasite. Toxoplasmosis you probably have heard of, most commonly associated with disease concerns in pregnant women. So we're going to spend a fair amount of time talking about why that's an issue and how that can be dealt with. Toxo is what's described as an obligate intracellular parasite or organism. Basically, this means that it cannot survive without cell mechanisms that are provided by the host. It requires the host for its own survival. This is a true zoonotic infection in that the actual parasite itself resides in cats species, all sorts of cat species, from our domestic cats all the way up to wild cat species. And it can have a range of intermediate hosts that it actually makes ill. So remember, reservoirs tend to not become ill. The intermediate hosts do. So it's everything from us, humans, to a variety of other animals. And then sadly, this particular parasite is one of the #1 reasons for death in sea otters along the California coast, which they actually believe is a direct result from cat feces being washed in wastewater into the seawater, and it's killing large numbers of sea otters along the California coast. So the way it tends to work is the cats shed the oocysts, and they do so into areas where they defecate. So sandboxes are a real issue. Gardens tend to be a problem. And so the actual oocysts remain present in the soil, sand, and even on the surface of vegetables potentially grown in the garden, if there's soil attached to them. So the primary ways it's transmitted is actually contact with cat feces of some sort in soil, but you can also get toxoplasmosis from undercooked meat. If, for example, there's another intermediate host that has the parasite present. And it can also be transmitted across the placenta. This is where the issue comes from with pregnant women. That is referred to as transplacental transmission. So before we talk a little bit about what the disease actually looks like, I wanted to discuss a little bit about the parasite itself and some of the forms of the parasite. So as we were discussing with crypto, there's more than one type of form. And so let's talk a little bit about the life cycle of this parasite. So first you have a cat reservoir, and it sheds the oocyst into, let's just say, soil. Then what happens is the oocyst is ingested by an intermediate host. In this case, let's say a human. Now, where you get a branch point here is what happens depending upon how immunocompetent the host happens to be. So let's say you have an individual that that's immunocompetent, meaning that their immune system is capable of handling the parasite. This means that they'll be asymptomatic, and essentially what happens is that as the parasite moves through its various stages, the immune system destroys it. So let's talk a little bit about the stages of the parasite. So first there's the oocyte, which is the ingested form, and the form that is excreted from the reservoir, the cat. Then you have what's called a tachyzoite, and this is within the oocyst, and it is the infectious form. That's the form that actually infects cells, and it also happens to be the form that is capable of being destroyed by medication. So it's the treatable form. The tachyzoite then becomes a bradyzoite, which is the reproductive form. And these remain within the cell. Therefore, they are not treatable with medication. So what happens is if you have a host that's immunocompetent, that has a good immune system, then the immune system destroys the tachyzoite form, and the cells do not become heavily infected, which means the person is asymptomatic. If someone's immunocompromised, then the immune system does not destroy the tachyzoite form, and cells are infected by the bradyzoites. So you have a spread of infection. So in people that are immunocompromised, this parasite can actually cause some pretty severe symptoms, very, very high mortality, and encephalitis or inflammation of the brain is a real problem as well. If it happens to cross the placenta, then the fetus is definitely immunocompromised, and it can result from everything from miscarriages, fetal death, premature infants, to what this picture is representing, which is hydrocephaly, or water on the brain, which can be a result of encephalitis in the womb. So let's talk a little bit about the risk to pregnant women. So the thing that's sort of interesting, if you want to phrase it that way, about toxoplasmosis is that it's only dangerous with the first exposure, because your immune system in general does such a good job getting rid of it. So the problem becomes if you have a pregnant woman who has never previously been exposed to the parasite, then they are at risk of acquiring it while pregnant, which makes their fetus at risk. If they have been exposed before, then the fetus is not at risk. So this is why, in general, they recommend that pregnant women just avoid any contact with cat feces. So you don't change the litter box. If you're going to do gardening, you wear gloves. There is tests that you can do to check for whether or not you have been previously exposed to toxoplasmosis. Like, interestingly enough, considering I've owned cats my whole life, I'm a veterinarian, I did this test when we were becoming pregnant, and I have never been exposed to the parasite. So if I had become exposed while pregnant, that would have put my baby at risk. So there are ways to find this out ahead of time so that you know for sure whether or not your particular situation and how risky it is for you. But the safest thing is just to avoid doing the cat pans, and frankly, who wouldn't want to pass that chore off? So that's what is recommended, is to avoid that and be careful while gardening. So if the actual diagnosis is done by serology, which we talked about earlier, And in this case, you're looking for particular antibodies, specifically what's called IgG and IgM. The IgG antibody represents chronic infection or previous exposure. It lasts in the body longer after exposure to toxo than does the IgM. Have an elevation in IgM represents acute, recent exposure. So the third option on discussion posts is to focus on toxoplasmosis. And we already talked a little bit about preventative measures, but I'd like you to go into a lot more detail about what can be done to try to prevent this disease, both in humans, not just pregnant women, but all types of situations. And then also what can be done to prevent this condition to other animals as well. The last one we're going to discuss is salmonella, which is probably one of the most common zoonotic conditions that you will run across. So what's interesting about salmonella is there are so many different variants or serovars of this bacteria. A lot of them have animal reservoirs. Some of the serovars result in what are called asymptomatic carrier states. These are individuals that have no symptoms but can pass along the disease. I'm going to need to pause here.